Assalamu alaikum students. The objectives of today's lecture are to learn about the various functions of kidney, general organization of renal structure, renal blood supply, and the structure of nephron. A large amount of metabolic waste products are produced in the body. Out of various organs, the renal system has maximum excretory capacity. The renal system includes a pair of kidneys, ureters, urinary bladder, and urethra. Functions of kidney. Kidney causes excretion of urea, creatinine, uric acid, and products of hemoglobin breakdown, metabolites of various hormones, toxins, and foreign substances like pesticides, drugs, and food additives. Kidney causes regulation of water balance by conserving water when it is decreased and excreting water when it is in excess in the body. It causes regulation of electrolyte balance by retaining sodium if the osmolarity of the body water is decreased and excreting sodium when osmolarity is increased. Kidney causes regulation of arterial pressure by regulating the volume of extracellular fluid through renin-angiotensin mechanism. It causes regulation of acid-base balance by excreting acids and regulating the body fluid buffer stores. Kidney can also cause hemopoiesis. The kidneys secrete erythropoietin, which stimulates the production of red blood cells by hematopoietic stem cells in the bone marrow. Kidney can cause secretion of various hormones, including erythropoietin, thrombopoietin, renin, 125 dihydrocholecalciferol, and prostaglandins. Kidneys can also cause gluconeogenesis by synthesizing glucose from amino acids and other precursors during prolonged fasting. Physiological anatomy of the kidneys. General organization. Each kidney of the adult human weighs about 150 grams. The medial side of each kidney contains an indented region called the hilum, through which blood vessels, lymphatics, nerves, and ureter passes. The kidney is surrounded by a tough fibrous capsule. If the kidney is bisected from top to bottom, the two major regions that can be visualized are the outer cortex and the inner medulla regions. The medulla is divided into 8 to 10 cone-shaped masses of tissue called renal pyramids. The base of each pyramid ends in a papilla, which projects into the pelvis, which is a funnel-shaped continuation of the upper end of ureter. The outer border of the pelvis is divided into pouches called major calyces. These extend downward and divide into minor calyces, which collect urine from tubules of each papilla. The walls of the calyces, pelvis, and ureter contain contractile elements that propel the urine towards the bladder. This figure shows the general organization of the kidneys and the urinary system. You can see the minor calyx, major calyx, papilla, renal cortex, renal pelvis, renal medulla, and renal pyramids. Before we start discussion of individual structures of kidneys, let's first study the renal blood supply. The arterial system of kidneys includes a renal artery entering the kidney through hilum, branching to form segmental arteries, interlobar arteries, arcuate arteries, interlobular arteries, afferent arterioles, and glomerular capillaries, where large amounts of fluid and solutes, except plasma proteins, are filtered. Efferent arterioles. The distal ends of the glomerular capillaries form the efferent arteriole. The glomerular capillaries are the only capillaries in the body that drain into arterioles. Efferent arterioles form a second capillary network called the peritubular capillaries that surrounds the renal tubules. Therefore, the renal circulation is unique in having two capillary beds. Number one, the glomerular capillaries, and number two, the peritubular capillaries. The peritubular capillaries are arranged in series and they are separated by the efferent arterioles, which help regulate the hydrostatic pressure in both sets of capillaries. High hydrostatic pressure, about 60 millimeters of mercury in the glomerular capillaries, causes fluid filtration, and lower hydrostatic pressure, about 13 millimeters of mercury in the peritubular capillaries, permits fluid reabsorption. I repeat again, high hydrostatic pressure causes fluid filtration in the glomerular capillaries, while lower hydrostatic pressure in the peritubular capillaries permits fluid reabsorption. So, roughly saying, filtered plasma in the glomerular capillaries enters the nephrons and it leaves the nephrons by reabsorption into peritubular capillaries. Now, by applying your knowledge of physiology of circulation, it should be easy for you to understand that by adjusting the resistance of the afferent and efferent arterioles, kidneys can regulate the hydrostatic pressures in both glomerular and peritubular capillaries. Another important structure of the renal blood supply is the vasa recta. The tubular portion of the juxtamedullary nephrons is supplied by specialized capillaries called vasa recta. Juxtamedullary nephrons are one of the major types of the nephrons, which we will study a little while later, and they are supplied by specialized capillaries other than the peritubular capillaries known as the vasa recta. The vasa recta also arise directly from the efferent arteriole of the juxtamedullary nephrons. The vasa recta have a peculiar structure. They form hairpin loops. So there are two functional parts. Number one, the descending vasa recta, and number two, the ascending vasa recta. The descending vasa recta have non-fenestrated endothelium. There are no pores present in their endothelium, and they contain facilitated transporters for urea. And the ascending vasa recta, they have fenestrated endothelium. Now, both peritubular capillaries and vasa recta drain into the venous system. The venous system of kidney starts from the peritubular venule, draining into interlobular vein, which drains into arcuate vein, draining into interlobar vein, then the segmental vein, and finally into the renal vein. The upper panel of this figure shows section of kidney showing the major blood vessels. The lower panel shows the vascular supply of a nephron. This figure shows the relation between blood vessels and the different renal tubular segments. It also shows cortical and juxtamedullary nephrons, which I mentioned earlier are the two main types of nephrons. Also have a good look at the peritubular capillaries and the vasa recta. Both of these arise from the efferent arteriole, as I mentioned in the previous slide, but there is a difference of location and arrangement around the nephrons. Again, in this figure, note that the peritubular capillaries are present in the renal cortex and are more coiled in structure, while vasa recta extend deep into the renal medulla and form hairpin loops, having descending and ascending parts. Having discussed the important features of renal blood supply, let's now start the study of the nephron, which is the functional unit of kidneys. Each kidney in the human contains about 800,000 to 1 million nephrons. The kidneys cannot regenerate new nephrons. Therefore, with renal injury, disease, or normal aging, there is a gradual decrease in nephron number. Based on the location of renal corpuscle, which is a part of the nephron, the nephrons are classified into two main types: cortical nephrons and the juxtamedullary nephrons. The cortical nephrons are superficial. Their corpuscles are situated in the outer cortex, while the juxtamedullary nephrons are deep and their corpuscles are situated in inner cortex near the corticomedullary junction. Each nephron comprises two parts. Number one, Malpighian or the renal corpuscle, which may be situated in the outer cortex near periphery or in the inner cortex near corticomedullary junction, and it is the location of the renal corpuscle which divides the nephrons into the two types which I mentioned in the last slide. The second major part of a nephron is the renal tubule. Let's first study the structure of renal corpuscle in detail. Renal corpuscle is formed by glomerulus and the Bowman's capsule. Glomerulus. Glomerulus contains a network of branching capillaries with afferent arteriole on one end and efferent arteriole on the other end. The glomerulus is encased in Bowman's capsule, and filtrate fluid flows into Bowman's capsule and then into various parts of the tubule. The endothelium of glomerular capillaries is made up of single layer of endothelial cells. It is fenestrated, having pores, and it is attached to a basement membrane. It also contains the mesangial cells, which are stellate cells located between the basement membrane and the capillary endothelium. The functions of the mesangial cells are following. They are contractile, play a role in the regulation of glomerular filtration. They secrete the extracellular matrix. They can take up immune complexes, and they are involved in the progression of certain glomerular diseases. Note the mesangial cells located between two capillaries in this figure. Also note the podocytes. I will explain in a short while what are podocytes. The second part of renal corpuscle is the Bowman's capsule. It is formed by two layers: inner visceral layer, which covers the glomerular capillaries, and number two, the outer parietal layer. Now pay attention to the next few slides. Both layers of Bowman's capsule are composed of a single layer of flattened epithelial cells resting on a basement membrane. And this basement membrane of the visceral layer fuses with the basement membrane of glomerular capillaries. Therefore, the basement membranes, which are fused together, form the separation between the glomerular capillary endothelium and epithelium of visceral layer of Bowman's capsule. The epithelial cells of the visceral layer of the Bowman's capsule are fused by or formed by the cytoplasmic extensions called pedicles. These cells with pedicles are called podocytes, as I showed in the last figure. And these pedicles are arranged in an interdigitating manner, leaving small cleft-like spaces in between. The cleft-like space is called slit pore. Now have a good look in this figure and note that how the podocytes form filtration slits on the basal lamina and the relation of the basal lamina to the capillary endothelium. I stop here to end today's lecture. We will continue with the structure of nephron in the next lecture.